Anybody want to come sit in the front row? It's empty now. They weren't able to make it in today. Too much rain. It's nice. Okay, quick review. We've talked about meiosis before in class. So if you start with a diploid germline cell, it undergoes meiosis and produces, let's talk male just to make it easy, so we don't have to deal with polar bodies and the oocyte. Let's just consider males for a minute. If you have one stem cell, one diploid germ cell that undergoes meiosis, how many cells get produced? How many sperm? How many? One meiosis. 4 cells get produced. So if you're capital A over capital A, if that's the genotype of the diploid cell, then what are the haplotypes of the 4 sperm that are going to be produced? You start with one cell, and it undergoes meiosis, and you produce 4 spermatids. What are their haplotypes going to be? All A. Everyone is going to have a capital A. Why? Because the parent didn't have anything else. The dad's genotype was, what do we call that when you've got the same allele on both chromosomes? Homo something. Homozygous. Okay, so we've got 2 homozygous genotypes up. See, all together, we can do it. We've got 2 different homozygous genotypes up here, and the one in the middle is heterozygous. So what is the product of meiosis from the other homozygote, little a over little a? 4 sperm get produced, they all have little a. Because that parent only had little a alleles, they can't put anything else in their haploid gametes. What's the point of all this? What happens in the middle? What does the heterozygote do for the first time? If it's a heterozygous dad and he produces 4 sperm, this is Mendel's first law. If you're diploid, and you know it, no. Sorry, I was about to break out in song. Half of the gametes are going to get capital A's, and the other half will get lowercase a's. That's Mendel's first law, the law of segregation of alleles. It's all about meiosis, the production of sperm and the production of oocytes. If you're a heterozygote, you've got a capital A and a lowercase a. The capital A goes into half of the gametes, the lowercase a allele, the other sister chromatid, by definition, goes into the other half of the gametes. And this doesn't just happen in this one meiosis, it happens every time one of those cells goes through meiosis, which means it's not just 2 capital A's on the left and 2 lowercase a's on the right. It's when you look in the gonad of the individual, there may be, especially if it's a male producing sperm, there could be 10,000 sperm that have capital A, and the other 10,000 sperm are going to have lowercase a. Guaranteed. It's Mendel's first law. Occasionally it gets broken, but we're not going to deal with the exceptions to Mendel's first law. So this is the basis for all of Mendelian genetics and really transmission genetics. So remember, today we're going to talk about Mendelian genetics. Some next class, and we'll get into pedigree analysis. It's our 2 concepts in Mendelian genetics. It's really all about being able to predict, based on genotype, the phenotypes of individuals, and to be able to predict, say you wanted to be a genetic counselor and you had a husband and wife or a man and woman or whatever you want, to call them, come in and say we'd like to have a kid, and the genetic counselor has to look at the genetic histories of the families and the disease history of the families and be able to counsel that couple on, should you have kids? What are the probabilities that your kids might have these familial genetic diseases that run in your families? But before we get there, one last point about translation, if you want to hear about it. I never talked about this point on the bottom right. How did scientists determine that codons are 3 nucleotides long? If you're curious, I'll tell you. Okay, so we'll work through it quickly. How did scientists determine, how did geneticists determine that ribosomes read 3 nucleotides at a time instead of 4, instead of 2, instead of 6? It's pretty straightforward. what they did was they synthesized messenger RNAs that, say, contained all the same letter over and over again, like U, 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 U. They fed that to a ribosome that bound to it. And so what would this ribosome, now we know that codons are 3 nucleotides long, what polypeptide would that ribosome produce, assuming it had a start codon? It would produce phenylalanine, because there's one phenylalanine codon, a second phenylalanine codon, a third phenylalanine codon. But that would work if the codons were 4 nucleotides long as well, right? 4 U's could encode phenylalanine, the next 4 U's could encode phenylalanine. So the next thing that the scientists did was to vary that RNA that they synthesized and fed into the ribosome. So instead of just all the same nucleotide, you start to add a little bit of nucleotide variation, like UUC, 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 UUC. What would that produce? Also phenylalanine over and over and over again. But the point is, by varying the nucleic acid sequence, that would be F, 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 just like above. But what would have been the case if the ribosome was reading every 4 nucleotides? What if codons were 4 nucleotides long? The first one would be UUCU. That would encode some amino acid. But what would the next codon be?
UCUU.
UCUU, and then it would be CUUC.
Those are all different.
Those would be all different, or presumably those would be 3 different amino acids. Let's call them X, Y, and Z, although we know that Y actually stands for tyrosine. So what they did was they started playing with different variations of different sequences, and they discovered that it was only when they had repeating units of 3 that they got the same amino acid encoded over and over and over again. If it was reading every 2 nucleotides, that would be UU, and then CU, and then UC, and then UU. They would have expected to get 4 different amino acids in the protein. And that's how they figured out that if it was only when they varied every 3 nucleotides, they got the same amino acid over and over again. All right, so Mendelian genetics. Boil it down a little bit. We're just going to talk about one type of cross, the monohybrid cross. And the goal, as I mentioned earlier, is not only to be able to organize genotype data in a Punnett square, which is what you have to do to be able to do the second task. If you have a cross and you know the genotype of the parents, the whole idea is to be able to predict the genotypes of their offspring and the phenotypes of their offspring. So that's going to require us to know a little bit about meiosis, which we already talked about at the start of class, a quick refresher, and also about dominance. So we'll see if we get to dominance today. We might start covering that next class. We'll see. So before we get there, I'm going to tell you a story about an experiment that happened in my lab. It involves the green fluorescent protein, which is a protein that, it's a protein, so it's encoded by a gene, the GFP gene, which was isolated decades ago from a jellyfish. And the structure of this protein means that it absorbs light of a particular wavelength and then it emits a green fluorescent light. It's a really cool protein that's been used in lots of different labs around the world, which is why in 2008, the scientists who initially extracted this gene from jellyfish and designed it for use in genetic studies won the Nobel Prize in medicine. So pretty much everybody uses GFP in one way or another. Biochemistry laboratories, cell biology laboratories, genetics laboratories. And I have a particular strain of worm in my lab that has this GFP gene inserted into it. So worms don't normally glow green, they're not jellyfish. But this particular strain of worm has the GFP gene from jellyfish. It's been slightly genetically adapted so it works better when expressed in worms. So that gene will produce that GFP protein, but it only does so in the pharynx of the worm. So here's a picture of the head of a worm under just regular light, and then what it looks like under fluorescence. So just its pharynx produces this green fluorescence. Those are the only cells in the worm where GFP is produced, and that's because the GFP gene, there's the gene, right? What's upstream of the gene to the left? The coding region on the right side upstream of the +1 nucleotide is the promoter. And in this case, somebody has put a promoter that they knew causes that gene only to be expressed in pharyngeal cells, so in the throat of the worm. And this is what we call a marker gene. So if you wanted to know where the throat of the worm was, you put a green fluorescent protein upstream of, or downstream of its a promoter that we know expresses proteins only in the pharynx, and you can see where the pharynx is. So we can have chromosomes that have had GFP, the gene, inserted. Right? Worms don't normally have this gene, which means that that is a transgenic worm. It's had a gene from another species, trans, put into it. So no chromosomes in worms naturally have the GFP gene, which means we're going to have two alleles. We'll have an allele, versions of this chromosome that have the GFP gene, and ones that don't. So I'm just going to refer to those as capital G for the allele that contains GFP and a 0 for the versions of the chromosome that haven't had that gene from jellyfish inserted into the chromosome. Okay? Any questions about that? So is this worm homozygous or heterozygous for GFP? If a worm had those two alleles in every one of its cells? Heterozygous. It's heterozygous. One version of the chromosome has the gene, the other one doesn't. In other words, you don't have to be heterozygous doesn't mean you have to have two alleles of the gene. You can also be heterozygous if one chromosome contains a gene and the other one does not. So a normal cell doesn't have that gene at all. Both of its alleles don't have GFP, so its genotype is 0 over 0. It's homozygous for not having GFP. So then because worms, like humans, are diploid, you can have 3 different versions of genotypes. You can have the worms that are homozygous not having GFP in the bottom right. You can have heterozygotes, where, like we saw above, one allele has the GFP gene, the other one doesn't, and you can have homozygotes that have both copies of the chromosome have that transcriptional unit, that gene. So which of those 3 genotypes will produce green glowing worms? These 3. First and second, top 2. Does anybody disagree?
The first one for sure, but the second one we don't really know.
So that's the question is, if you don't have GFP, this 0 over 0, you're definitely not going to be green, so you don't produce the GFP protein. So the question is it enough to have one gene producing the protein, or do you have to have both alleles producing the protein? What do you think?
I think it's enough just to have one.
Well, one allele produces green fluorescent protein, which glows when it's exposed, fluoresces when it's exposed to a particular wavelength of light. So yeah, both of these genotypes, whether you have 2 copies of G or just one, make GFP, and those worms are green. Now the one that's homozygous G, G over G, may be more green, might be brighter green, it might produce more green fluorescence because it's got twice as much protein, because there are 2 chromosomes, each of which have this gene. So maybe those worms are making twice as much green fluorescent protein. But all of those worms have that GFP gene and they'll be green. So the question is, and this is what happened in my lab, we had 2 heterozygotes that we wanted to cross together. Here comes Mendelian genetics. We want to know what's going to happen in the next generation. So in that line there in the middle on the left, what generation is the first generation of a cross called? It's a notation that geneticists use. A letter and a number. These are the parents. So the letter is P. P0 generation, the parental generation. So just by definition, the first generation across we call the parental generation. It's denoted P0. I don't know why it's just not just P, but we'll go with it. And then their offspring are which? F1, which stands for the first filial generation, which is a Latin word filius, meaning brother. So it's the first offspring generation, F1. And then after that, if the F1s have kids, then that's the F2 generation, and F3, and so on. So we wanted to know what the F1 worms would look like if we crossed two heterozygotes. And presumably, both of these worms are green. So here comes Mendelian genetics. We wanted to predict, I would like you to help me predict, what are the F1 worms going to look like? Are they all going to be green or not? So we need to get some background information first before we do that. So we already looked at what happens when a G/0 cell, a heterozygous cell, undergoes meiosis. That cell will produce 4 daughter cells, gametes, after the 2 rounds of meiosis. And what are their haplotypes going to be? From a heterozygous somatic cell here that undergoes 2 divisions of meiosis, produces 4 cells. What are the haplotypes in each of them? We've got G's or we've got nothings? Okay. So like we saw at the very start of class, half of the gametes are going to get the G, and the other half get 0. Why are there 4 chromosomes? From the diploid cell, how is it that we produce 4 gametes and there are 2 chromosomes that have G here and there, and there are 2 that don't have the G?
Because the first division, they duplicate the DNA.
Right. So there's synthesis before meiosis, so then we get temporarily a gamete that has 2 G's and 2 zeros, and that's why then all 4 of those, each chromosome goes into one of the 4 gametes, as we've seen before. And again, that really is Mendel's first law. With replication of chromosomes and then segregation of each of the alleles, 1 per gamete, 4 chromosomes go into 4 gametes, those are the 4 that are going to be produced from a heterozygote. There are going to be 2 different types of gametes, half G, half no G, normal chromosome. And the same is true here from the mom, the bottom. She'll produce 4 gametes. And because the mom, that's male, isn't it? M, male, produces the same. Half of his gametes carries G's, the GFP transgene, and the other 2 don't have GFP. Bless you. And then what does the Punnett square, or the Punnett axes, reveal? Why do we use it? What is it? Why do we decide to draw some sort of figure to represent what happens in the next generation?
It's probability.
It's representing frequencies, probabilities. So as I indicated in the video, what this is that's indicating if each of these axes stops at 100% of gametes, that is, there are 4 gametes, 1, 2, 3, 4, GG, 0, 0. How many of them have G? Half. So that's what that distance is representing, half of the gametes. So think of each axis as a frequency of the gametes that are produced. Half of the gametes have G, the other half of the gametes have 0. So what the Punnett square really indicates is what happens when random fertilization events occur. Males make all of these sperm, and the hermaphrodites, or females, in C. elegans or C. briggsii, or in any other species, produce oocytes. And fertilization is random most of the time. That's another example of where Mendelian genetics sometimes goes awry. So we always assume when we're talking about setting up crosses that there's random fertilization with an infinite number of gametes. In other words, this is an ideal situation where what the geneticists imagine is the kind of experiment where you take an infinite number of oocytes and an infinite number of sperm and throw them all into a tube, and fertilization would happen randomly. which is true in vivo as well. When fertilization happens naturally, that's also true. There's a random chance of one sperm of whatever genotype, haplotype, encountering and fertilizing an oocyte. So in other words, the axes of the Punnett square represent the frequency of, in this case, male haplotypes on one axis, and the frequency of female haplotypes. Remember, the haplotype is just the genetic constitution of the gamete. And in this case, it was from the male, half G, half 0. And from the female, it was also 50% carried G and 50% don't have the transgene. So axes represent the haplotypes of the gametes produced by the P0 generation. That's the last thing to remember. So what's in the middle of the Punnett square? What's in the square? What does it represent? Yeah? Who said that? It was right. It's the F1 what?
Haplotypes?
So this represents, for example, G, let's see, female is on the y-axis. So a G-carrying oocyte can encounter one of two types of sperm, either a G or a nothing. So we can get G over G or G over 0 with those fertilization events. So what are those genotypes? Talking about what happens after fertilization. What gets produced after fertilization from parental gametes? What's produced? A zygote. Of which generation?
F1.
F1. So that's the other thing to remember about Punnett squares, is that what's in the square itself are the F1 genotypes. So all this is a graphical representation of fertilization and what happens after fertilization. We get the genotypes or the haplotypes of mom's gametes and dad's gametes, and the many different possible ways they can come together at random to produce different genotypes in the F1 generation. Any questions? Why is it a Punnett square and not a Punnett rectangle? Is there ever a time where you could have a Punnett rectangle? So what makes it a square?
Because there's 2 on the bottom and 2 on the side.
She beat you to it, sorry. Right, we're diploid, so we only have the possibility of producing 2 different types of gametes, 2 different haplotypes of gametes. What would this look like if this was a salmon that are tetraploid? What would a Punnett square look like for a salmon? Tetraploid, a salmon has the possibility of having up to 4 different alleles of every gene. So then you would need a 4x4. And we're not going to do this, by the way, don't freak out. We're going to do strawberry. We're going to do 12x12 Punnett squares. And you have to. But this is the concept, is that that's what the Punnett square represents. It represents the different types of gametes. So a salmon might have 4 different versions of a single salmon. A single individual have 4 copies of every chromosome, could have 4 different versions of a gene. So you'd have to draw a bigger Punnett square. So here's maybe a trick. Here's another way to not get a Punnett square. You could do a Punnett rectangle instead, if this helps. Maybe it doesn't, but just an alternative. What does the Punnett square for this cross look like? Let's say it doesn't really matter where the axes are, but so if we put let's say, the male on the y-axis and the female gametes on the x-axis, how would we fill the rest of that in? How many different haplotypes does the mom produce? What are they? What sorts of gametes? So she'll produce 4 gametes, and they're going to be half capital A, half lowercase a. What about dad? Homozygous. How many different types of gametes does he produce?
4 all lowercase a.
They're going to be all, he produced 4. They're all going to be lowercase a. So, traditional Punnett square, dad produces lowercase a at the same frequency he produces lowercase a, and mom has half capital A, half lowercase a. Do we really need to have both of the lowercase a's on the y-axis?
No.
Because that axis represents, so I heard a no. What does that axis represent again? Each axis represents the frequency of something. Frequency of the different haplotypes. But if there is no different haplotype, why not just, you don't have to do this, but for those of you that don't like Punnett squares, maybe this helps. You don't even have to write the 2 different gametes on there, because dad doesn't make 2 gametes. The reason that we write the 2, getting too excited, hang on. The reason that we write the 2 different gametes side by side is only really for heterozygotes, because we need to track that there are 50% capital A's and 50% lowercase a's, because this axis just represents the different types of gametes. If you've got 100% lowercase a from dad, why divide it up? You don't need to. There's no biological need to do that. So what are the different genotypes of the F1s? When you fill in the Punnett square. Heterozygous and homozygous. I heard the R word, which is a word we're going to talk about a lot. Recessive. Why did you think it's recessive? Because it's lowercase. We're going to talk about that. So we'll come back to that point. But it's fine to use recessive for now. So half of the offspring are heterozygous, because half of the area of the square is heterozygous. So again, the square is just a graphical representation of, as was pointed out earlier, frequencies. The numbers of individuals that have different genotypes in the next generation. So in this case, the square is half capital A over lowercase a, and the other half of the area is lowercase a over lowercase a. Any questions or clarifications? Okay, so for that same cross, G over 0, G over 0, what are the genotypes we get? Fill in the Punnett square. So what are the ratios? There's a genotype ratio and a phenotype ratio. That is, each of these genotypes, we already predicted what the phenotypes of the worms with these different genotypes are going to be. What were the phenotypes? If you're capital G over capital G, you're green or not? Green. So fill it in with some green. What about G over 0? They do have one GFP transgene, so we're going to say they do produce GFP and they are going to be green, and we decided that 0 over 0 is not green. They don't have the GFP gene, they're not going to produce the green fluorescent protein, they're not going to be green. So that means that for every 3 green worms, we expect to observe 1 not green worm. There's a worm there, but it's not going to be GFP, it's not going to be green. That's the prediction, that's what we use Punnett squares for. Set up a cross. We want to know is what we observe in the next generation, what we expected to observe. This is our expectation. that's the phenotype ratio. Because we're talking about a phenotype, a trait. We're not talking about genotypes yet. So 3-to-1 phenotype ratio. How does that relate to the genotype ratio? What are the 3 different genotypes we have? We've got capital G over capital G. We've got capital G over 0, and we've got 0 over 0. So there's, in this cell, in this Punnett square, there's 1 G over G, 2 G over 0, and 1 0 over 0. And you've probably seen this too many times, but there you go, one more time. 2 different ratios that you expect from this type of cross, which is called a monohybrid cross. In which mono means we're looking at one gene or one locus, GFP or not GFP in this case, G or 0. And hybrid in this case, what does that mean? Well, it does mean cross, but in this case, genetically, hybrid means heterozygous. In other words, what we're looking at in a monohybrid cross is there's a heterozygote at a single gene, and we're crossing it with another heterozygote for that same gene. That's what a monohybrid cross is. Heterozygous, 2 heterozygotes crossed together. And then that's when you see these 2 different ratios, a 3-to-1 phenotype ratio. There's an expectation here that we're going to talk about when we start talking about dominance, though. And a 1 to 2-to-1 genotype ratio. And to be really straightforward, it's those one individual that has the G over G and the 2 that are G over 0 that together make 3, which is the same as that 3 and the 3-to-1 ratio. Right. There are 3 cells up here that are individuals that have this gene and will be green, but they're made up of 2 different genotypes. There's 1 capital G over capital G, and there are 2 that have GFP, but not on both alleles, not on both chromosomes. So that's how the phenotype ratio derives from the genotype ratio. 1 to 2-to-1, genotype becomes normally, at least in this case, 3-to-1 in phenotype. So that's how those ratios are related, according to the question at the bottom of the slide. So to reiterate, the goal of using Punnett squares is to make a prediction about what we expect to observe. And we're going to expect and observe, which means we're headed once again toward a chi-square test. So Mendel's first law, segregation of alleles into gametes. That's the prediction that if you're heterozygous, half of your alleles become one allele, the other half of your gametes get the other allele. So here's the weird part about the story. We already predicted what happens when you mate 2 heterozygotes. Draw it out again just for completeness. We expect that 1 to 2-to-1 genotype ratio with 3 of the worms being green to every 1 worm that's not. We've got green worms, 3 of those for every 1 that's not green. But when it's crossed in my lab, this is what we saw. So we got 14 worms, 3 of them, we genotyped them. So we had, how many of these worms are green? 13. So we saw 13 green worms and 1 not. Is that a 3-to-1 ratio? That's a 13-to-1 ratio. So then we wondered, there are two possibilities. Either that just happened by chance, we picked the wrong 13 worms and they all happened to be green, or this means that Mendelian genetics is not occurring properly and we're not actually getting Mendel's first law. We're not getting independent segregation of alleles at gametogenesis. So to figure that out, the question is a 3 to 10 to 1 ratio statistically significantly different from what we would expect, assuming that Mendel's laws are working in this case? So what are our expectations in this case? What are the expected numbers of worms with those 3 different genotypes that we should have seen? So we should have seen a ratio of 1 to 2 to 1, but that's a ratio. That doesn't tell us how many individual worms we would see of each of those 3 genotypes. So how many worms should we expect? See, we observed 3 worms that were G over G, and 10 that were heterozygous, and 1 that was totally lacking the GFP gene. So what are the expected numbers of worms? We had 14 total worms observed.
7 of them should be heterozygous.
So a 1 to 2 to 1 ratio, I think what you're getting at is that means that 25% of the worms, 50% of the worms, and 25% of the worms should be those 3 genotypes. It's 1 to 2 to 1 ratio, 25%, 50%, 25%. And so absolutely, if we observed 14 worms, we expected that half of them, 50%, should be G over 0. That means that number is 7. If Mendelian genetics are working, and we should have observed, half of, right, 3.5 and 3.5. So are they significantly different or not? And that's why I distributed to you at the start of class a Google Classrooms exercise. If you'll fill in this information and conduct the chi-square test, that's worth 2 points, due by the end of class. Actually, I'll change the deadline. I'll make it the end of the day. So please work on that at some point today, turn it in. I'll give you 5 minutes now, and then we'll close with a couple other points about dominance. So if you can get through it all right now, that's good. If not, you can have until the end of the day to finish up. If you've got any questions, please ask. Come around and answer questions if you have any. Dear friends, this is the most misunderstood topic in genetics. And here's why. And this is why we talk about this. So we're going to talk about, these are just vocabulary. There will be some tasks associated with dominance and recessiveness at some point, but this is the basics. So this is what we're going to talk about. Pure breeding, recessive traits, dominant traits, and the misleading use of capital and lowercase letters to indicate dominance. It's true, it's a system that geneticists use. Normally we use capital letters to indicate dominant alleles and lowercase to indicate recessive alleles, but that's not always true, and that leads to some problems sometimes. Does anybody in here have brachydactyly? You don't have to tell me if you don't want to. It's a pretty common trait. I know a few people that have brachydactyly, short fingers, short toes. This is caused by a dominant mutation. It is known, it has been known for decades, now people are checking their fingers. People have known for decades that a dominant mutation in a gene causes this phenotype. Is that surprising to you, and why would that be surprising to you? We've got how many people in this room? We've got like 50-ish. Presumably, at least no one's told me they've got brachydactyly. So if it's dominant, why don't more of us have brachydactyly?
I'm actually not surprised because I'm assuming that since, ecology happens, that it was preferred by people who are mating to have longer fingers, which is weird, probably saying it out loud, but that might...
This comes up every time I teach genetics. Go for it. Yeah.
It might be something like sexual preference.
Okay. So maybe more brachydactylous people mate with other brachydactylous people, so it's in a small percentage of the population, maybe. Other thoughts? Yeah.
Isn't it because, like you said, it's dominant, so you're saying why it wouldn't be more? Because you would assume if they're heterozygous, then you would have it. Like the minimum requirement would be heterozygous, and everybody would have it. But more people are homozygous recessive for this allele. And I don't know the specifics. But
Yep. So these together, I think, are the critical points. There may be some aspect of human-based selection on who you decide to have kids with. It's up to you. No one's judging anybody. But this is the really critical point, is just because it's dominant, if you remember anything from this class, because you're going to move on, is eventually, most of you, to evolution, if not all of you, it's really incumbent upon me to make sure that you're using dominant and recessive in the correct fashion. Please, please, please remember this, if nothing else. Dominance is not equal to a very closely related word, which I think is what causes all the confusion. Because it's dominant doesn't mean it's common. Dominance does not mean it's the wild type, that it's the most frequent or the most common trait. It just means that inside the cell, the dominant allele has the major effect in the cell. So it turns out absolutely, brachydactyly, it is caused by a dominant mutation. But it's not a very common mutation, so very few people have that mutation and very few people are brachydactylous. This is part of a fantastic but very old school sort of stiff upper lip genetics debate from decades ago. This is the only thing I'll ever read verbatim to you in a class. To the editor of Science, this is a scientist writing to the prestigious journal Science, I am reluctant to intrude in a discussion concerning matters of which I have no expert knowledge, and I should have expected the very simple point which I wish to make to have been more familiar to biologists. But some remarks of Mr. Udney Ewell, to which Mr. Punnett has called my attention, suggest it may still be worth making. In other words, what are you stupid geneticists talking about? There's no reason. So what Ewell had said down here at the bottom was that if brachydactyly is dominant, in the course of time, over generations, you would expect to get 3-to-1 phenotype ratio. 3 brachydactylous to every 1 normal. And the gentleman who's writing this, William Hardy, of Hardy-Weinberg equilibrium, which we're not going to have time to talk about, says there's not a slightest foundation for this idea that dominant mutations will spread. That is, that they'll become more frequent. Dominance does not equal predominance. Just because it's dominant doesn't mean it's going to be in more and more and more people as time proceeds. So I'm going to end with this argument, and then I'll tell you what we're going to be up to next class. What I've drawn up here are a bunch of different matings between individuals. So each genotype is meant to represent the genotype of an individual. So we have 10 pairs of individuals up here. Assume these are humans, and there's one individual up here that's got brachydactyly. Who's got brachydactyly? There's one individual, there's only one capital B in this whole slide. And I've used capital B to illustrate that it's dominant, capital letter. So if and there are 10 pairs, which means there are 20 people, PPL, on this slide. So what's the frequency, the phenotype frequency of brachydactyly in this population of 20 people? How many people have it?
1.
How many total people?
20.
20. So this is 5% frequency. What's going to happen? Time to make predictions. So before that, though, the bottom 2 bullet points. Brachydactyly, because capital B is dominant, could occur if you're homozygous for capital B or heterozygous. And you're only wild type if you've got what genotype? The recessive genotype, homozygous recessive or homozygous little b. So that means 19 of the 20 people up there are wild type. They don't have brachydactyly. There's one brachydactylous individual. So to complete this then, let's assume that every one of these 10 pairs has 4 kids. 4 is useful because it's Punnett square-ish. So let's pick on one of those crosses, of which there were 9 on this sheet. What are their offspring going to look like genetically? Do a Punnett square. Punnett square. 100% frequency of capital B from one of the or lowercase b from one of the parents. 100% frequency of lowercase b coming from the other parent, so all their kids are going to have to be also homozygous recessive. So that means that we get, and I will wind up, that means from every one of these pairs we get 4, 8, 12, 16, 20, 24, 28, 32, a total of 36 lowercase b over lowercase b. If everybody's having about the same number of kids, and most of the couples in the population are homozygous recessive, they can only make more wild type homozygous recessive kids. So to end, what's the Punnett square for the one family that has brachydactyly?
Like 50/50.
Mom produces only lowercase b's. Let's assume that's mom. Dad has half capital B, half lowercase b. Oh, and they were having 2 kids, or 4 kids. So we've got 2 capital B over lowercase b, 2 lowercase b over lowercase b. So in the whole population, that means there were 36 little b over little b plus 2 from that couple. And what other genotypes and how many? There were 2 heterozygotes. So the heterozygotes are brachydactylous. The 38 are wild type. So 2 out of 40 individuals are brachydactylous. The population frequency of brachydactyly is? 2 in 40 is 1 in 20 or 5%. Didn't change from the previous generation. That is why it's the frequency of the mutation that matters, but if everybody else is normal and they're all having normal wild type kids, then the frequency of the trait doesn't change. It stays the same, 5% every generation. So please take a look at see what to do for next class. We'll finish up a little bit on dominance. Yeah, the purebred dog. We'll do a little bit of dominance next class and we'll talk about pedigree analysis. Thanks for hanging out.